I think yes. Estás en directo. Yes. Okay. Here, Pierre. Hi, Pierre again.
Hi again.
Hello, good morning.
Good morning.
Hi.
How are you? Well, good afternoon.
Yes.
Yeah.
So Beatrice also is here.
Hello, Beatriz. Nice to meet you.
Hi, nice to meet you.
Nice to meet you, Pierre.
Nice to meet you too.
So we can wait maybe 2-3 minutes more and we can start, I think, no? Laura, what do you think?
Yes, we can wait 5 minutes
Okay.
till 12:05,
Mm-hmm.
just in case there are any problems to have access. And we
Okay.
are already on streaming,
Yes.
aren't we?
Yes,
Okay.
we are.
Okay. Let's wait a little bit.
Okay. Good afternoon, Manuel. We are also in Cadiz. Thank you for greetings from Cadiz.
1 minute to go.
Yes.
Okay.
Okay, let's get started, no?
Okay. Thank you very much, Maria. I think we can start our session, CU Talent webinar session. Welcome all. I'm very glad to have you here today. As you know, CU Talent webinars is an initiative of a research project, which is an AIDS 2020 funded project that belongs to the CU Alliance, Alliance of European Universities. With this initiative, which is led by the University of Cádiz, we are trying to communicate, disseminate the research that is being performed by our junior researchers in our alliance. A project in which different universities are involved, the University of Western Brittany, the University of Kiel, the University of Split, the University of Malta, and the University of Dansk. Today, we are going to talk about food security, topic which is getting more and more interest by researchers and by society. When we think about the 1950s, 1960s, if we think a little bit about that period, we're not, our researchers or society were not concerned about how anthropogenic activities could provoke an impact in the environment and in human health. More and more different researchers started to identify substances that were produced by anthropogenic activities that could produce harm, could produce a negative effect to our environment. We started with rivers, and now more and more we heard about effect of different contaminants or different products of anthropogenic activities that could provoke adverse effect in the marine environment. And when starting with the marine environment and freshwater environment, researchers started also to identify that there were different products, different substances that are being produced by anthropogenic activities, or sometimes even natural activities that could produce effect to humans, could affect negatively to human health. This is a topic that is more and more studied nowadays and is of high concern for researchers and for regulation, for regulatory frameworks. It is more and more important to identify substances, anthropogenic substances or natural substances that can produce adverse effects, not only to the environment, but also to the ecosystem. When I talk about environment, I'm talking about ecosystem, not only to the ecosystem, but also to human health. Today, we have two presenters, two researchers from the University of Western Brittany in France, one of our partner universities. And they are going to show a different point of view or a different face of of food security. Beatriz Arce López, she's a chemist, and she's working in chemistry, biochemistry, biology, and is going to focus her presentation in food security and that point of view. And on the other hand, we have Pierre-Yves Cantelain, a researcher also from the University of Western Brittany, that is going to show us another point of view. Is going to show us the view of food security, taking into consideration political sciences and public relationship, which is, I mean, his, let's say, his base knowledge. So I would like to start with Beatriz Arce López. Beatriz Arce López is a postdoctoral researcher at the Laboratoire Université Laboratoire Université de Biodiversité et Écologie Microbienne of the University of Western Brittany, de la Université de Bretagne Occidentale. And she's going to present her research that deals with mycotoxins and the problem of their presence in food and in food safety, and which one is the interest for human health. So she's going to make a presentation entitled Mycotoxins, FB1 and OTA, Toxical Effects on Human Cells. New in situ models. She's not only going to show us how these mycotoxins could affect human health, but also she's going to show us new approaches in vitro experiments. So I would like to give you the floor, Beatriz. You have 20 minutes, and then we will go with Pierre and some questions if people are interested in asking you something about your research. So I'm giving you the floor, Beatriz. Thank you very much. It's very nice to meet you.
I hope you can see my screen.
Yes, Beatrice, we can see it.
Okay, so thank you very much for the kind introduction and all the organizers of CU Alliance that make possible these kind of seminars. It's my pleasure to be here today as part of the speakers. As mentioned, I'm Beatriz Arce, and I'm going to present a talk related to food security and the relationship with my postdoctoral research carried out as a Bienvenue Marie Curie co-fund postdoctoral fellowship in the Laboratoire Université de Biodiversité et d'Écologie Microbienne in Brest University in France. So today's agenda will include an introduction about food security and what are mycotoxins, and my research project MiBitox, and finally some take-home messages. So when we hear about food security, the question is, what do we think about it? So first of all, I would like to introduce this concept that has been defined by the Food and Agriculture Organization, FAO, of the United Nations, that food security exists when all people at all times have physical, social, and economic access to sufficient, safe, and nutritious food that meets their dietary needs and food preferences for an active and healthy life. So that means that, on the other hand, food insecurity is the limited of uncertain availability of nutritionally adequate and food safe, or limited of uncertain availability to acquire acceptable foods in socially acceptable ways. So food insecurity does not necessarily cause hunger, but hunger is a possible consequence of food insecurity. In this context, it's also important to mention the relationship between food safety and food security. The challenge of food security is to assure that all people have access to enough food to live a productive life. But a large part of food security is assuring that food is safe from chemical, biological, and physical hazards. And there are other numerous aspects of food security, including the effects of climate change. That is going to be a future topic. Food safety is therefore an umbrella term in which are included all the hazards during handling, production, and storage of food. So how does food safety help achieve the Sustainable Development Goals? By 2030, the idea is that everyone, in particular the poor and more vulnerable population, have access to safe, nutritious, and sufficient food. So we focus mainly on those two Sustainable Development Goals, number 2, zero hunger, and number 3, good health and well-being. Food safety also contributes to economy-related and other goals. Food can become unsafe at any point of the supply chain, and contaminants may enter to the food commodities, affecting food safety and with an impact on human and animal health. As we can see in the image, among the potential contaminants, mycotoxins are of particular concern due to the importance in terms of food and feed safety. So you will think, what are those mycotoxins? So mycotoxins are toxic metabolites produced naturally by several fungi, and they can occur at different points of the food chain, whether storage or in the field. And there are different mycotoxins already known. But to get better insight of such an important topic, the scenario encountered in the study of mycotoxins is similar to that of an iceberg. Most of the studies tend to focus on the free mycotoxins, and what happens with the others. There are a large number of compounds that might be of interest of food safety and that will need to be included to understand the risk that they pose to humans. So mycotoxins reach people by different routes, being the most important ingestion, either through the direct consumption of contaminated raw materials such as cereals or derived products, or indirectly through the consumption of food derived from animals that have been contaminated through their feed. In general, those mycotoxins are really stable molecules that once they enter to the food chain, they are difficult to be eliminated. So exposure assessment is usually performed by two different approaches. The first one is by analyzing those levels in food and link this data to consumption data, which is known as external exposure. Another approach is the analysis of the biomarkers in biological matters, such as blood, plasma, or serum, or breast milk, or nails, hair, or others. And in this second approach, I have been published a previous work, which is considered about human biomonitoring in plasma, blood, and serum from human samples. in human mycotoxin risk assessment, the two different approaches are going to be considered. So first of all, I would like to focus on the backgrounds, considering the analysis of food. As we know, the human diet is very varied and complex. The heterogeneous distribution of these compounds in food and the difficulty to obtain accurate consumption data. And also we have to think about the lack of knowledge of the viability of these compounds in humans, also taking into account that food processing is could modify the food. So on the other hand, human biomonitoring or internal exposure represent an effective strategy to assess the risk of these compounds, because we have to think about the inclusion of all sources of exposure. Also, we about to collect and analyze a large number of food and human consumption data, and it really evaluates the human exposure. So we can think that those two strategies are seen as complementary approaches in the analysis of population of risk to mycotoxin exposure, although the second one is seen as a strategy on the rise. In the present and in the future, new dietary habits such as more plant-based diets, or such as flexitarian or vegetarian, or the consumption of pseudo cereals such as quinoa, are showing higher mycotoxin contamination levels due to the increasing exposure. Also, worldwide climate change has been identified as a driver of food safety issues because of the increase in temperature and CO2 concentrations in the air, as well as the change in precipitation levels impacting the mold development. So even the most studied mycotoxins are already regulated at European level, we have to think about it that the reality is that one fungus can produce more than one type of mycotoxins, so the most likely scenario corresponds to multi-mycotoxin. And those compounds can affect human health in many different ways. As is well known, one fungus can produce more than one type of mycotoxins, so co-exposure and co-occurrence of several mycotoxins will be included. As already mentioned, the co-occurrence of mycotoxins can result of different synergetic, additive, or antagonistic effects. So the question is, does this multi-mycotoxin contamination modify individual effects, and in which sense? And to get better insight of those links, in toxicology, we try to develop new cell culture models closer to the reality to in vivo in order to assess the toxicity. Those studies, and in our group has been published a previous review focused on in vitro toxicological approaches to evaluate the toxicity of such mycotoxin mixtures. So in this context, it's been focused my project research, which is called MIBITOX. So mainly the objective of this project is been to study single and combination of those two mycotoxins, ochratoxin A and fumonisin B1, which are already regulated, and we know more or less their individual effects, but not in combination. The idea is to evaluate different innovative in vitro models, 2D and 3D, in order to simulate more the reality and compare the toxicological effects of those mycotoxin mixtures on different hepatic and colon adenocarcinoma cells in different states. So first of all, a systematic review has been focused to fill the gaps in knowledge about the effects of mycotoxins, ochratoxin A and fumonisin B1, on human in vitro systems, focusing on intestinal and hepatic cell models. As we know, the first barrier of detoxification are those organs, so it was important to focus on them. So the review has been focused on those three parts: the occurrence and co-occurrence in cereal and cereal-derived products in Europe, the ADME studies, which means the absorption, distribution, metabolism, and excretion, and the toxicological effects. This work will be submitted soon for publication. So I will try to explain you a little bit what is included in review. Cereals and its derived products are the first food resource in the world and represent a huge, important or concerning issue worldwide in food and feed. But the thing is that in Europe, we are mainly protected thanks to different European alerts and legislation. First of all, we have the RAS, which is the Rapid Alert System for Food and Feed. And beyond the legislation, this tool has been established to ensure the exchange of information between the different member states and the food safety authorities in case that a risk appears to health and it's going to enter to the food chain. So they communicate quickly in order to avoid those risks to human population. And according to our review, we have seen that the European Commission legislation pointed out that the levels in cereal and derived products for ochratoxin A has to be 5 micrograms per kilogram, and for fumonisin, 1,000 micrograms per kilogram. So here we have focused and we have shown the RAS notification during the last 10 years in Europe in different countries. As we can see, there are some values that have exceeded those levels, so they had been rejected for direct human consumption. There's also in Europe a really important European Food Safety Authority, which is based in Italy, and the main objective of this authority is to provide scientific advice and carry out risk assessment on the wide range of chemicals. So mainly member states collect data on occurrence, on contaminants in food and feed, and EFSA coordinates the data collection with the different member states with a useful system which is called Foodex, and we can obtain all the data from the different member states. Also, scientists, we focus on the risk assessment, and in order to work with the European Union risk managers, EFSA provides scientific advice to support and to take decisions about the safety of these substances for human and animal health. So in this context, the idea is to evaluate the human exposure to multiple mycotoxins of interest, mainly ochratoxin A and fumonisin B1, which are considered as possible toxic contaminants by the year, and causing co-exposure and different toxic effects, being the liver the main target organ. So most studies have been carried out in vivo, so in animal experiments, but the idea of our project is to develop new in vitro models from the most simple to more complex and realistic models. And the first barrier is the intestinal barrier, and then also the hepatic cells are considered in toxicology as a good cell lines because the liver is the seat of metabolic conversion of food and toxic molecules. So that's the importance of studying those different cell lines. To date, there's still very few data on chronic mycotoxin exposure of those cell lines. So the purpose of this project has been to seed those cells individually on 96 well plates and in co-culture in trans well plates in order to evaluate the cytotoxic effects, performing different tests, mitochondrial activity for 2D cultures and ATP for 3D spheroid cultures. In addition, using inserts and with the TIR study, we performed the study of the potential bioavailability of eucratoxin A and fumonisin B1 by stimulated mycotoxin transfer through the intestinal epithelial barrier in aqueous conditions. All along this project, mycotoxin concentration and potential metabolization were monitored using analytical studies thanks to the use of liquid chromatography coupled to mass spectrometry cutoff in extracellular and intracellular fractions. And this project is still ongoing, so hopefully in the following weeks or months, we'll have the results already published. So finally, I would like to give you the last take-home messages. And first of all, we have to think that there is no food security without food safety, that the importance of studying food safety, and because of that, mycotoxins are an important food safety challenge, as we can see. And the new climate scenarios and new dietary habits are important because they will modify the levels of those mycotoxins and the exposure to human population. And the importance of using more realistic in vitro models, such as 3D or organoids, in order to simulate the exposure to those mycotoxins and to reduce the use of animals in those studies. And I would like to result the significance of this project to the assessment of the risk associated with human exposure to mycotoxins. This project has received funding from the European Union Horizon 2020 Research and Innovation Program under the Marie Slobodska-Curie Grant Agreement. And I would like also thank you all my supervisors and my lab colleagues, and also the CU Alliance for this conference, and all of you for hearing. And finally, I would like to introduce you an important project which it will be set up in my lab, and one of my supervisors is in charge of this project. And it's important for the general population. It's a participatory science project called MINION, which means mycotoxin migration, and it's funded by the French National Research Agency. And its main project is to identify the risk scenarios and provide recommendations to reduce food waste for the general population. So if you are interested in participating, you can just have more information in this website, or you can contact me or my supervisor, and we count on you. Thank you all of you for your attention, and I will be grateful to answer finally some questions. Thank you very much.
Thank you very much, Beatriz. What a nice presentation. You start with the beginning of mycotoxins and the origin, and explaining everything very clearly to all of us that maybe we are not specialized. I would say that I'm not specialized in this topic. It's not my topic of research, but I was able to understand everything. I have a couple of questions. I will leave them for the end, but I would like to congratulate for the presentation and for these taking home messages, that I think are very useful. And I will do later on some comments regarding, I guess that is a citizen science project, what do you talk about at the end, isn't it? Yes.
Yes.
Okay. So.
Yes.
Okay, so I will do some comments later on. Thank you so much, Beatriz.
Thank you.
And now is the turn of Pierre-Yves Cantalan. I don't know if I'm pronouncing it in the right way. For me, Beatriz Arce López is easier.
It's perfect.
Okay. You are, or he is a researcher as the postdoctoral researcher at the CRBC Center at the University of West Brittany. And your background is political science and public relations. His talk relates very well, or his research relates very well, food security, climate change, and energy transition, what I think is very important and of concern. He's going to present today his research that deals with, or is entitled as methanation, I don't know if I'm pronouncing in the right way, methanation units development in Brittany, conceptualizing the links between food security, climate change, and energy transition in the Anthropocene. So, I mean, a great interdisciplinary research. So the floor is yours, Pierre. Thank you very much.
Yeah, well, thanks a lot. Sorry, I have some difficulties to put my screen on. Well, thanks a lot for the invitation to this seminar. And firstly, let me present a little bit my research perspective, because I've made my PhD about the international politics about Amazonia, as far as political science is concerned. And basically, I was really interested in the notion of environmental commons as a notion that could describe the Amazonia, the tropical forests, and, well, that can also open to new fields of research and to new objects of studies. And what I'm going to present to you today is the result of my former postdoctoral research. I was actually, you were right to say that I was in the CRBC, but now I am on the other side of Brest at another lab, which is called AMUR, about the deep sea. And well, the general coherence of my research is precisely to study different kinds of environmental commons so as to provide a general framework about power relations around those environmental commons. And of course, it has a link with food security when we talk about this second object of study I chose, which is the methanization development in Brittany, but actually it's not only developing in Brittany, but that was my field of study for interviews and for observation. But this development of methanization has an impact on, of course, agriculture. Just let me present you what is methanization first. Well, basically, methanization is a biogas or electricity production process going from fermentable material, and that also needs a complement of carbon, which is, well, most of the time, like the fermentable material basis in Brittany is livestock manure, and the carbon complement is corn. And so those elements, of course, strongly interact with the agricultural model that is at stake in Brittany. And per se, it also has an impact on environmental commons that I define as those spaces whose destruction or fast alteration may threaten large parts of the living and human species life conditions. Because of course, as we're talking, like climate change and the threats, mass extinction of the species are, well, basically remodeling all the considerations about the political relations of power around environmental commons. And so in Brittany, the methanization was well, particularly supported by a journal that you can see here on the screen called Britain Peasant, Le Paysan Breton. And this journal is basically financed by, well, important companies in the agroalimentary sector of Brittany. And is also like supportive of the position of the dominant union, which is called the FNSEA. And if you read the article, this article is basically advocating for development of methanization given the context of the Russia-Ukraine war, because we have a dependence on the Russian gas, right? And so the Paysan Breton was advocating for basically the lowering of the norms that were concerning the methanization, so that actually we can produce more biogas in Britain. And of course, it's quite interesting to have to know that this sector of agriculture in Brittany is supportive of methanization, because methanization, as it produces biogas, is of course linked to the energy transition, and is most of the time, let's say, positively coined as well, green gas, as positively evaluated dimension of the energetic transition. And in political science in France, there is some kind of emerging group of studies about what we call green extractivism, which means like extractivism that is precisely happening in the name of the energy transition. And that was one of my angle of analysis about methanization in Brittany, because of course, like many environmental comments are concerned when we talk about agriculture in Brittany. We can think about, of course, the atmosphere because of the greenhouse gas emission. We can think about the surface waters, which are basically 80% of waters in Brittany that are exposed to the certain mode of agriculture, the soil, of course, and the biodiversity conditions. So my study is about the insertion of the methanization development within on the one side, the world economy, and on the other side, its impacts on environmental commons. So those are the two main lines of analysis. And first, well, the first conclusion, and that is going to be published in an article in NetStream. The first conclusion is that basically the development of methanization in Brittany strengthens the intensive breeding industrial filière, I don't know how to say that, filière, like industry. The breeding industry is strengthened by the development of methanization. Well, first, you can see on those maps that the first one is about breeding pigs, the second one about broiler chicken, and the third one about egg-laying poultry. Well, you can see that on those matters, Brittany is the first region in France, the first producing region in France. And that's also what actually explains that in most cases, methanization is based in those kinds of exploitation because they are the dominant farming production in Brittany, though actually the livestock mania that come from these farms are quite weak in methane, which is kind of a paradox. But actually, precisely that paradox can be explained by by a political and economic issue that I'm heading to. And you can see that here on this table that is made out from official data. Well, you can see that the livestock mania is the most important source for the methanization units, and no matter actually which kind of methanization is concerned, being at the farm, being collective, or being centralized. Centralized is more like urban one, etc. But in any case, like in Brittany, the most important source is livestock mania, despite the fact that it is weakly, it doesn't contain much methane. So why would that? How can we explain that? Well, basically, the thesis I defend is that the development of methanization strengthens the intensive farming industry, and it's actually going to perpetuate it in a way. And I want, like, as an example of that, I want to take a conference that took place in December 2022 with the CEO of the Brittany Ferries about the future of the industry in Brittany. And the Brittany Ferries is a company that was basically created by some wealthy peasants of northern Brittany. And this company is projecting to engage the energy transition of its boats by using methanization processes. And this, the CEO of this company is still really linked to the intensive farming industry in Brittany. So the fact that there is a transition, green transition, which is organized from this sector in Brittany, actually produces a perpetuation of the intensive farming dependence of the Brittany economy. Because why would that create a dependence? Well, basically, if you develop a methanization and that the methanization is based on livestock manure, as I've just proved, well, then this circle is gonna will need to be fed, basically. And so you raise the demand from for organic material by developing methanization in Brittany. So in a way, the fact is that those very peasants that are developing methanization right now are considering themselves as pioneers, and they are pioneers of the energy transition and so on. But if we look at other works in agricultural studies, well, basically you see that this imaginary of agriculture as trying to pioneer and so on, well, is really linked to a linear conception of agriculture, and that in this conception, methanization would only be one more step on the global progress of agriculture since the so-called Green Revolution, which was not that green. And so, like this is like how also we can frame the development of methanization in Britain. So that's the first conclusion. Methanization, as it is being developed right now in Brittany, is actually perpetuating the intensive farming and intensive breeding model, which is, of course, quite adverse to environmental commons. If you take the greenhouse gas emissions factor, or if you take the quality of water, or if you take, well, another example that leads me to my second part, which is the international dependence of this production. Because it's important, once we've said that this methanization development is going to perpetuate the intensive farming, it's also important to see how then the development of methanization is inserting itself in an international political economy dynamic. Because if you take again, like the argument of the journal I was introducing you to, the Paysan Breton, if you take the argument upon which it was defending the methanization, well, basically the argument is that it's going to make France and Brittany more autonomous regarding the energy production, like in the context of the war. But the fact is that if methanization production is relying on the intensive breeding, the intensive farming, well, then it is also relying on the importation of soy, for example, to feed the animals. So to produce your biogas in Brittany, you need to import soy from Brazil, for example, and then you would have some effects of what we call imported deforestation. So if your soy is imported from Brazil and that this soy needed some part of the Amazonian forest to be cut, well, then it also adds another dimension to the relation between methanization in Brittany and the environmental commons, because you're threatening, by perpetuating this model, you're contributing to the threatening of the very existence of the Amazonian forest. So that's the first dimension. And then the, well, more biological dimension is that if you rely on this production, then when you import soy, you import also like nitrogen. And then, well, this excess of nitrogen is basically what's going to pollute, what's going to contaminate afterwards the waters in Brittany. So the insertion is both economical and ecological. The insertion of methanization is both economically and ecologically adverse to the environmental commons. And actually, this reality, this socio-political reality, is reconducting, is reproducing some quite old oppositions in Brittany between models of agriculture. Because since the development of intensive farming, well, you've always had some critical voices that were pleading for an alternative model and so on. But and those also defend the pursuit and the continuation of this model based on peats and intensive farming. And basically, which is quite fun, and that was also a part of what interested me in the study of methanization here, is that the current defenses of intensive farming were really in favor of methanization, of this green energy tool, transition tool. And the defenses of the alternative were opposed to that, which is totally different from any other socio-ecological conflicts in Brittany. So that was an interesting situation. And my work, well, basically explains that this converse situation, the fact that the dominant agricultural, the dominant peasant union is in favor of methanization, is explained by the fact that methanization is gonna perpetuate the model they defend, while actually the other union that you have here in yellow, the Peasant Confederation, well, they're in favor of a moratorium on methanization. So this is basically just reproducing the difference between two conceptions of the agricultural model of development in Brittany. And current development is, of course, more in favor of those who basically dominated the agricultural policy in France since a few decades. And the methanization, the development has to be read from this socio-political reality that is basically also a food security challenge, because when you presented, Beatrice, you showed us like this nice scheme, well, saying that environment was a clear component of food security. And basically, well, if you threaten the environment, well, you threaten food security. And that, well, I was reading this summer the works by the Indian biologist Vandana Shiva, or there's the works by a French economist, Hélène Torgman, and both are basically raising the issue that the intensive agricultural model is, if you project in the longue durée, well, this model is basically threatening the reproduction of a sustainable agricultural model. So, well, that was the point of my demonstration, to show that even if it is inscribed in the green transition, the methanization development is not necessarily akin to environmental commons, and that might be even analyzed as adverse to environmental commons as it is now reproducing the intensive farming model, which is quite problematic in Brittany. Thanks a lot for your attention. We can't hear you, Maria.
Yeah. Oh, okay. Thank you so much. Yeah, I was saying that thank you so much for your presentation, for congratulations for your research and for giving us this point of view of methanization and the socio-political dimension of methanization. It's very interesting. It's very interesting piece of research, and I really appreciate this contribution, since it's very interdisciplinary. I mean, interdisciplinary research, and it's not a local research at all. You need to take into consideration and to have a global dimension to interpret all your results. I really find it very interesting. So thank you so much. And now we are going to go to the questions, if there are any questions. Maria, could you have a look to the chat if we have any questions?
I don't see questions. No, I see one. Marta Motsrevska asked, congratulations, very interesting. Why exactly Fumonish B1 and orcharotoxin? Beatrice, it's for you. I'm sorry if I pronounced it incorrectly.
No, it's okay. Ochratoxin A and fumonisin B1. So the idea is that among the 300 mycotoxins that have been identified, only around 40 are already regulated. So during my PhD, I've developed and studied 19 different mycotoxins, and I try to analyze, I analyze more than 400 human plasma samples, and in them I saw that ochratoxin A appears in most of the samples. So that's why we decided to choose at the beginning this mycotoxin, because it's already regulated. It's a potential carcinogenic mycotoxin, and it's important for human health. And on the other side, fumonisin B1 is considered a mycotoxin as an emerging mycotoxin. As I explained before, we can see that the free mycotoxins, but also they are emerging mycotoxins, which will be important and are important for the future. And so this mycotoxin is also considered as a possible carcinogenic. And the idea was to combine both of them, because there are not a lot of studies till now, and both they are important for human health. So we could have chosen others, but for example, aflatoxins, they are already known as carcinogenic for human health, and there are a lot of studies in both of them. So we decided to choose those important mycotoxins. Thank you for the question.
Thank you so much, Beatrice, for the answer. I cannot see more questions in the chat. So probably somebody would like to make a question by voice.
I have some questions. I don't know if there is anyone else. So if there is not anyone else, I will go ahead. I have a question for Beatriz. I'm working on my topic of research is ecotoxicology, marine ecotoxicology, and how contaminants of emerging concern affect invertebrates, marine invertebrates, and we are measuring, we measure different biomarkers stress, of DNA damage, and also we perform different toxicity tests with these contaminants of emerging concern to find out how they affect reproduction and mortality in early stages organisms. But I was thinking about this, about your research, and for us it's very difficult sometimes to demonstrate how, I mean, our results regarding the adverse effects of contaminants at the molecular level, how this effect could be extrapolated to the ecosystem in general. I was wondering if these in vitro experiments and the results that you obtain performing these in vitro experiments, how are they extrapolable? How can they be extrapolated to human health, to human itself? How we go, if it is easy on your topic of research to go for interpreting in vitro results to human health itself. I don't know if I explain myself in the right way, Beatriz.
Yeah, I think I understood, but I think I understood your question, but otherwise you can interrupt me and add some comments. So yeah, basically the aim of the project is to see the toxic effects on human cells. So the idea is to go from the most simple model, which is the 2D, and go to a really complex model, which will be the organoids. So seeing the effects in 2D from 3D, we see that the model is more sensitive and we can simulate better the effects on the human body, because as we know, cells are not like flat. So we try to grow them in order to see how they are going to be in our body. Also, the development of co-culture models, which allow us to put on the same plate. On the one side, we put the intestinal and the hepatic cells, try to simulate how the intestinal barrier could protect the hepatic organs or others. The idea is to go from the most simple to the most complex in order to simulate what's going to happen in our body and to reduce the use of animals, because there have been a lot of studies in this topic related to animals, but now we're trying to reduce it. So in vitro models are such an important technique in toxicology in order to see what's going to happen in our body and not to be exposed directly human population to mycotoxins, because we are already exposed through ingestion. So we need to see how it's going to be due to all the different exposure routes, as I already mentioned. I don't know if I answer.
Yes, perfectly. You answered it perfectly, and I understood very well what you wanted to say. I got a little bit surprised in the presentation myself. I got surprised myself when you were talking about mycotoxins and the food in which is present. No? Now that we are all, in this kind of old-fashioned diet where, I don't know, soya is like, they recommend soya, and they recommend dry fruits like almonds and so on. Many of the most recommended let's say it's food or yes, that is now in fashion, is included in this list of possible containing mycotoxins. So it's something that, I mean, it's of great concern. And I really hope that the research that you are performing has not only an impact at the scientific level, but also at the society level, and that it can be transformed in new regulatory frameworks for food safety or food security. As you say, there is not food security without food safety, no? So it's very, very interesting. And my other question is not really a question, it's just a comment that at the end of your presentation, you mentioned a citizen science project. So, and you already talked a little bit about it. We are in Research U project, we are disseminating citizen science projects or research projects through our social media and in the webpage. So, we will contact you because we have Beatriz Díaz Garduño, that is a technical communication manager in our project, that maybe can help to disseminate the project, and maybe we can look for participants in all the alliances universities. So we will contact you on this regard, because we really find that it's very interesting. Okay, thank you so much.
Perfect. Thank you very much.
Thank you, Beatriz. And I have another question for Pierre. I find your, as I said, very interesting presentation. Most of all because of how you have focused methanization in this socio-political dimension. I would like to ask you about I mean, we know that methane is not only coming from agriculture or farming, let's say farming, but also there are other sources of methane in the atmosphere. And it's the more methane that we have in the atmosphere, the more increase in temperature we have, since one molecule of methane can warm much more than a CO2 molecule. So I was wondering, because now there are, I mean, maybe it's not your topic of research itself, but it's true that in the last decade, the concentration of methane in the atmosphere has increased a lot. And the reading is higher than 10 years ago. And I was wondering if you have, Pierre, if you have taken into consideration this in your research, if you know anything about this.
Well, yeah, thanks a lot for this question. Actually, the production of methane would be more linked to the intensive farming than to the very process of methanization itself that is going to produce gas and so on, and it's producing gas by, if you focus on the production of methanization, the production process, then you have, of course, a reduction of greenhouse gas emissions compared to natural gas. But then if you integrate the other dimensions that I've discussed, well, this conclusion is not that clear. Because of course, like the intensive breeding is a huge producer of methane. And as you said, methane is even more problematic for climate change, inducing more climate change than carbon dioxide. And there's also the dimension about nitrogen. Because if you put too much, well, that's a global statement, but still, that's one part of what Anthropocene is now. Like we've put so much nitrogen in the soils that actually the cycle of nitrogen is strongly altered And so, well, there's a concentration of, in French, we say protoxide d'azote. I don't know if it would be nitrogen protoxide, maybe, in the atmosphere. And those have a power of a climate change that is 300 more than the power of carbon dioxide. So, of course, like, well, my research is raising the question about the relevance of inserting a technique by supposing it is going to be neutral, while actually if you insert that in an already existing economic dynamics that is hostile or adverse to climate change dynamics, well, that is positively feeding it, well, then you're going to feed it also. I don't know if my answer is properly answering you. But
Yeah, thank you so much. Thank you, Pia. Yes. Well, so are there any more questions? Okay. Well, I have to say, Pia and Beatriz, that this is in the streaming, that you can find the videos in YouTube in our CU Alliance channel. That all of you, I mean, if you are interested in contacting these great researchers, you can go to our webpage and you can also take our social media. I encourage you to have a look to what we are doing and the dissemination that we are performing, all of you. I would like to thank, first of all, the speakers, the postdoctoral researchers. Thank you very much for your time and for doing such great research. And I also would like to thank Maria Yancombaya and Beatriz Diaz, our technical managers that are behind all these Zoom issues and dissemination and preparing the webinars and the agendas and so on. And finally, of course, to our attendees, I mean, the researchers, PhD students that are attending the webinar, thank you very much. So with these words, I don't know if our technical managers would like to say something else. Maria?
Thank you, Laura. Also, I would like to thank you for leading this meeting, our CU Talent, for our researchers who provided with this very interesting presentations, and to our participants. Thank you so much, and we will provide you with the certificates of the participation during the month. I don't see any more questions in the chat, and nobody asked. So you can write them to our researchers, the questions, if you have more, and I think we can finish. Thank you so much to everyone.
Thank you so much. Have a nice day.
You too. Bye-bye.
Thank you.
Bye. Thank you.
Thanks a lot.
Thank you.